Hello Brainerds, my name is Jace and welcome to the Big Brain Busters. So in this video, I'm going to look at the gram-positive cocci. So this is part 1 where I'm going to look at the pathogenic Staphylococcus species and we're going to classify them, and then mainly we are going to jump into Staphylococcus aureus. And as usual, I've also added some practice questions at the end of this video to see whether you've understood. With that being said, let's jump into it. Okay, to look at the divisions under gram-positive cocci now, we can look at them using the catalase test, of which we have two under this category. We have the micrococci, which are catalase positive, and streptococci, which are catalase negative. Our main focus for this video are the micrococci, which are further divided into four classes. We have micrococcus, stomatococcus, and planococcus, of which these guys are non-pathogenic to human beings, so neglect them. Our main focus is Staphylococcus, which are further divided using the coagulase test. So there are coagulase positive Staphylococcus, which is there is only one, Staphylococcus aureus, and there is also coagulase negative Staphylococcus, or the cons, which are coagulase negative, which are Staphylococcus epidermidis and saprophyticus. Okay, let's talk about the Staphylococcus species now, but before we go to that, let's understand what the word Staphylococcus simply means. So Staphyl means a bunch of grapes and coccus means berry. So when you view these Staphylococcus species under a microscope, they look like a bunch of grapes stuffed together and in a shape of berries. So that's why they came up with the word Staphylococcus. Okay, and the species under this genera are Staphylococcus aureus, Staphylococcus epidermidis, and Staphylococcus saprophyticus. Okay, and in this order, this is the way they are pathogenic, having Staphylococcus aureus to be the most pathogenic. Okay, so now let's talk about Staphylococcus aureus. So this guy is a gram-positive cocci, catalase positive, coagulase positive, meaning that it is able to produce an enzyme known as coagulase, and it has a diameter of 1 micrometer. Then it is a facultative anaerobe, meaning that it lives in an environment without oxygen but can also survive in an environment with oxygen because it is facultative. Then it is non-motile, self-explanatory, non-sporing. Then it is occasionally encapsulated, meaning that not all strains of Staphylococcus aureus contain the microcapsule. Some do, some do not. Okay, then it is the most pathogenic cocci, and it can develop drug resistance, where we're going to talk about methicillin resistant Staphylococcus aureus strains. Okay, then on laboratory findings, you find that when you put it on nutrient agar, it is able to produce a golden yellow pigmentation, as you can see from my slide there, my diagram. And then another laboratory finding is that when you put it on sheep blood agar, it is able to produce beta hemolysis strains, okay, as you can see from that. Okay, so now to talk about the virulence factors of Staphylococcus aureus, we can divide them into three categories. We have the cell wall associated factors, the toxins, and the extracellular enzymes. So virulence factors are these guys which make Staphylococcus aureus to be pathogenic. Okay, so when you hear of virulence factors, just think of pathogenicity of an organism, a microorganism. So these are the guys, and you can follow my mnemonic, either TEC, CET, or ETC. And this is how I like following them because that's how I put them in my head. But either way, just do what works best for you. Okay, so now let's talk about the cell wall associated factors. I have a mnemonic to remember all these, and it is MTAP. So the M is for microcapsule, then teichoic acid, adhesins, peptidoglycan layer, and protein A. So you can use this mnemonic to remember all the cell wall associated factors. Here is a structure of the cell wall and its factors. We're going to discuss that later. And then here is another property, a special property for protein A. We're going to discuss that. Okay, so now let's talk about the microcapsule. Here is a structure of Staphylococcus aureus and the layers in that order. So remember that the outermost layer will always be the capsule layer or the slime layer, which is the blue layer there. And then we have the peptidoglycan layer followed by the innermost plasma membrane. Okay, so now remember that not all strains contain the capsule. Only a few strains have the capsule. And what's the function? It's simply for inhibiting opsonization by neutrophils. Okay, then this is the main function of the capsule. Then another function is simply it has a zwitterionic structure, which just simply means that it has both negative and positive charges, which allows it to form abscesses. So the zwitterionic structure is for abscess formation. So to talk about teichoic acid, there's really not much to talk about this guy because he just only promotes cell surface adhesion to the mucosa, and just like the microcapsule, it also inhibits opsonization. Then to look at it in the structure of the cell wall, as you can see, we have teichoic acid right over there, and these are the functions. Okay, so to talk of the adhesins, just as the name suggests, they function to adhere to mucosal surfaces and to themselves. Okay, so there are three main adhesins that I would like you to know, which are clamping factor, fibronectin, and collagen binding adhesins. Okay, so the main one that I would like you to know, mostly the culprit, is clamping factor. So with this guy, it is very important when it comes to nosocomial infections or infections acquired in the hospital. So let's say, for example, your patient came into your department for some 8 months ago, and maybe they had a catheter inserted into them, or they had, a surgery, and then next thing they just start developing chills, fever, and systemic bacteremia. So you might start thinking in lines of, Staphylococcus aureus, because of this guy, the clamping factor. So it has the ability to form biofilms. So you find that the strains of Staphylococcus aureus, they will start clamping to each other, adhering to the catheter or any medical device that you inserted into a patient, and the next thing they'll start now forming biofilms. So these guys here will now form biofilms, which is like a glycocalyx layer that contains polysaccharides and proteins, and then now they will just develop this drug resistance. Okay, so the best thing that you would like to do for such a patient is just simply to withdraw the medical device and cut it out, all right, or maybe even insert another catheter or something like that. So that's it about the clamping factor that I really wanted you to know, about. Now let's talk about another virulence factor under the cell wall factors known as protein A. This guy is able to inhibit opsonization, has anti-complementary and anti-chemotactic properties, and in addition can cause platelet damage. Talking of these properties like this merely is requiring you to just simply master them, but I don't want you to master them. I want you to know the actual mechanism of action on how protein A evades the immune system and causes this. Okay, so to talk about that, allow me to just brief you through the structure of an antibody. So we have this Y-shaped IgG antibody, which has here the Fab region on these arms. Okay, this Fab region is also known as an antigen binding region, where an antigen of a microorganism will bind, and then they will form an antigen antibody complex. That complex is able to activate the complementary system, and in terms of opsonization, we know that when it binds there, that will be opsonization taking place, and when opsonization takes place, we know that there will be what? Chemotaxis, because this antibody will have the FC portion where the immune cells will come and bind to after opsonization. And what's the main function of opsonization? Simply for destruction, all right, destruction, agglutination, and all other things such as phagocytosis. Okay, so now in this case, when it comes to Staphylococcus aureus, how does it enable itself to inhibit all these immune functions? Well, protein A binds to the FC region, okay, of IgG antibody, and leaves the Fab region where it is supposed to bind. So it will bind on the part where immune cells are supposed to bind and leave the part where it's supposed to bind. So, and in this case, since it has left that part, meaning that immune cells will deem these antibodies, trying to opsonize Staphylococcus aureus, they'll deem these antibodies to say they are not bound to. Okay, now let's talk about the peptidoglycan layer. This guy here is thick, right? It has about 50 to 100 layers. So these layers you're seeing right there, these are the layers we're talking about, and they are about 50 to 100. Okay, and then the thickness is about 15 to 80 nanometers. Remember, it's a gram positive, so meaning that it must have a thick layer. Then what's the function of this peptidoglycan layer? It's simply to maintain the cell wall integrity by offering what? Rigidity. Okay, then apart from that, the another function is simply to induce inflammatory response, and also has an endotoxin like activity. Okay, now let's look at another virulence factor, which is the toxins. So you can use my mnemonic to remember all the toxins of Staphylococcus aureus. You can use MEAT. All right, the M is for membrane active toxins. So under the membrane active toxins, in other sources you will find them being referred to as pore forming toxins. So these guys are hemolysin and leukocidin, which is also known as Panton Valentine toxin. All right, then we have epidermolytic or exfoliative toxin. You will mainly find exfoliative toxin, but I've also added another name in case your lecturer uses it. Then you have enterotoxin, and lastly you have toxic shock syndrome toxin. Now let's talk of the membrane active toxins, which are hemolysins and leukocidin. Let's start with the hemolysins. We have alpha, beta, gamma, and delta hemolysin types. Among these, only gamma and delta are able to cause lysis on human red blood cells, while alpha and beta cause lysis on other species such as horses. The most important one is gamma hemolysin, which has 3 protein fragments which act with leukocidin to exhibit their function. I'll talk about that later. Let's go to the leukocidin. This guy comprises of 2 protein fragments, which are the fast and slow. These 2 protein fragments act with the 3 protein fragments of gamma hemolysin, and then they form a synergistic action that carries out lysis on red blood cells and white blood cells. Remember, hemolysins act on red blood cells, leukocidin acts on white blood cells, but then they form this synergistic action. In other sources, it is known as synergohymenotropic toxin. Not really high yield to know, but anyways. So this Panton Valentine toxin can be expressed on methicillin-resistant Staphylococcus aureus strains. And these strains, why they are called that, it's because they have developed a drug resistance against penicillin derivatives such as methicillin and oxacillin. So these penicillin derivatives are simply beta-lactam antibiotics, and these methicillin-resistant Staphylococcus aureus strains have developed drug resistance against those, so meaning that even if you try to treat your patient with such drugs, they won't improve in any way. Let's say your patient comes and then you put them on oxacillin. After some time, you find that their symptoms are not improving. It's simply because the strains that you're dealing with have got resistance against that drug. So the best thing is to change your drugs. Then these toxins are also associated mainly with community-acquired infections. All right. Now let's talk of another toxin known as exfoliative or epidermolytic toxin. This guy is able to cause Staphylococcus coded skin syndrome. So it is just simply a widespread blistering and loss of the epidermis. This toxin has got 2 proteins, which are ETA and ETB. ET just simply means exfoliative toxin. Okay, so ETA is chromosomal heat stable, and ETB is a plasmid coded heat labile protein. And this syndrome is mostly commonly seen in neonates and children, and in children it is actually called Ritter's syndrome, and it is characterized by the flip symptoms. I like calling them the flip symptoms because the skin is flipping. Okay, so these flip symptoms are what we call fever, lethargy, irritability, and poor feeding. Now let's talk of another toxin known as enterotoxin. So entero meaning it's something to do with the GIT. So this guy is a heat stable and gastric acid resistant toxin. It is a super antigen, of which we'll talk about later, the mechanism of action of super antigens. Then it causes food poisoning because it deals with the GIT. How does it do so? It stimulates the vagus nerve and the vomiting center. Okay, and in addition it also stimulates intestinal peristalsis because it increases motility. So if it does so, meaning that it causes symptoms such as nausea because it stimulates the vomiting center, and also vomiting. Okay, and with that there is also diarrhea because it increases the intestinal peristalsis. And you know that where there is vomiting and diarrhea, you are losing a lot of fluids, so meaning it will lead to hypotension. And then since you've lost a lot of fluid, dehydration will also kick in. Then when we talk of incubation period, after getting the toxin, it's about 1 to 6 hours. This is a high yield point, so you must know it. Then after that, you find that 8 to 10 hours the symptoms will definitely resolve. Now let's talk of toxic shock syndrome toxin. So it is also a super antigen, of which I've said we'll definitely discuss that later, and it causes toxic shock syndrome. And patients with TSS, they usually present with fever due to this widespread activation of cytokines, which will go and trigger the hypothalamus to change the set point of temperature. Yeah, then causes diarrhea, it causes nausea, it causes vomiting, it causes abdominal pain and hypotension due to this massive pooling of blood outside the vasculature due to inflammatory response. Then it causes also conjunctival hyperemia due to stasis of blood into different organs, such as also the conjunctiva, and also it causes myalgia. Okay, so that's much about toxic shock syndrome. Now let's talk of the mechanism of action for super antigens. So remember, in normal antigen presentation, we have an antigen presenting cell which has got major histocompatibility complex type 2, and then we have the T cell which has got T cell receptors. So these are the receptors for the two cells, such that a normal antigen which has been processed by an antigen presenting cell will be presented on its major histocompatibility class 2. For what? For activation of T cells. So in this case, the specific T cells for that particular antigen will be activated. Okay, so that's in a normal immune reaction, such that the T cells that will be activated will be only a percentage of 0.01. Now, with a super antigen, it does not bind in this normal peptide binding groove. It goes to link the beta domain of the T cell to the alpha domain of the antigen presenting cell, and then with this it causes this widespread non-specific activation of T cells, such that when it does that, it forms a trimolecular complex between the antigen presenting cell, the T cell itself, and the super antigen. So this is the trimolecular complex. Then with that non-specific T cell activation, and then there is now this widespread production of cytokines, and these cytokines are the interleukin 1, interleukin 2, tumor necrosis factor alpha, interferon gamma, and interleukin 6, which will now lead to those symptoms that I had mentioned earlier, such as hypotension and hyperemia in different organs, such as the conjunctiva, such. Talking of our last virulence factors, which are the extracellular enzymes, you can use my mnemonic CHODS. C is simply for coagulase enzyme, which is the high yield factor that you must know. And then you have the heat stable thermonuclease, at least according to the text I'm using, they're saying only Staphylococcus aureus has this. And then we have other enzymes such as hyaluronidase, lipase, and protease. And then we have deoxyribonuclease, which is also only found in Staphylococcus aureus. And then we have staphylokinase. To talk of the mechanism of action for coagulase enzyme, it causes formation of blood clots. How does it do so? There is a reaction between coagulase and coagulase reacting factor in the plasma fluid, such that this reaction will lead to conversion of prothrombin into staphylothrombin, and then staphylothrombin will lead to conversion of fibrinogen into fibrin, and then fibrin will now lead to clot formation. Now let's look at the clinical manifestations of the infection caused by Staphylococcus aureus. So we have skin and soft tissue infections, which will present with abscesses and boils, cellulitis, folliculitis, and impetigo. Okay, then we have also musculoskeletal infections that will present with what I call the SOPA. So these guys are septic arthritis, osteoarthritis, pyomyositis, and abscesses. Then we have also respiratory tract infections. Mostly with these guys, they'll present with infections as opportunistic. In let's say, for example, the patient had a previous viral infection, and then Staphylococcus aureus jumped in as an opportunistic infection. Then we also have bacteremia, which, with its complications, will lead to valvular endocarditis, and also we can also have septic shock. Then we also have toxin-mediated illnesses, of which we've already discussed, things like toxic shock syndrome and food poisoning. Okay, so let's try to see whether we can answer the question that we have been provided with by applying the knowledge that we have been given in the video. And by the way, I won't answer these questions to the fullest, but I'll give you just ideas on how you can go about them. And then if you have any questions, just drop them in the comment box, then I'll definitely reply. The question reads, Johnson was put on an oxacillin antibiotic therapy after a minor procedure of inserting a venous catheter. Despite several days of treatment, his condition worsened, presenting with an elevated fever of 40 degrees Celsius, and began to have chills, increased breathing rate, and delirium. His doctor then ordered a lab culture, and gram-positive cocci in clusters were reviewed in blood and at the surgical site, and a golden yellow coloration on nutrient agar was also noticed, so resorted to empirical treatment as he suspected bacteremia and drug resistance of the bacteria, thus decided to remove the catheter and change Johnson's antibiotic therapy. Further results after 4 hours of biochemical test results showed a coagulase positive test. What strains of the causative agent could be causing Johnson's symptoms? So in this case, with all the characteristics that we've been provided with, we know that we are talking about Staphylococcus aureus. So the strain under Staphylococcus aureus is simply methicillin-resistant Staphylococcus aureus strains. Then why did Johnson not respond to the oxacillin therapy? So it's simply because the methicillin-resistant Staphylococcus aureus strains have gained the ability of drug resistance against beta-lactam antibiotics such as penicillin derivatives. And under penicillin derivatives, we have drugs such as methicillin. Under methicillin, we have oxacillin drugs. Okay, so since oxacillin is a beta-lactam antibiotic, so meaning that this guy cannot work against these MRSA strains. Why did his doctor decide to remove the catheter? We know that the catheter could have been a source of infection. When inserting it, it could have been exposed to Staphylococcus aureus before inserting it. So that's why removing it, you could be able to remove the biofilm that the Staphylococcus aureus would have formed to the around the catheter. Then number 4, what antibiotic therapy would be suitable for Johnson? So since the doctor decided to change Johnson's therapy, drug therapy, in this case, the antibiotic therapy that we could use for Johnson is vancomycin. You can use vancomycin. Then Number 5, briefly discuss the pathogenesis of Johnson's condition. So here you're just simply explaining how the symptoms for Johnson came about, how fever came about. We know that fever will come about because we're talking about a foreign body that would definitely result into, this release of cytokines, and then go to the hypothalamus, change the set point, and then definitely will lead to fever. Then apart from that, we know that the chills and increased breathing rate and delirium or confusion will come about due to what? Due to damage to different body organs. So you explain all that using the virulence factors of Staphylococcus aureus. All right. All right, it's been a long video, too much information. Otherwise, thank you for watching, and don't forget to like, share, and subscribe as you enjoy your day. Otherwise, we end this video here. Shalom, shalom.